after finishing my tenure track there, I went to the dark side, as they tend to call it here. I chose for a step to industry, if only because I thought that if I were to continue an academic career, and I was teaching at a technical university in Delft, I was teaching maybe 80% of the students for a career in industry. I thought, well, I cannot just teach them from whatever I read in the books. I need to have a little bit of hands dirty by then. So that is why I moved in 2007. I moved to a local company called DSM. It happened to have its research headquarters in R&D, also in Delft, so that was practical. But also, as Luc said, as a biotechnologist, it was my first choice of company to work for, because I think people who know me know I tend not to brag, so I wouldn't have used this title unless I really believe there's some truth to it. I'm not saying DSM is the best company in the world. I don't think it is. It is not the most profitable. It's not the biggest. But I do think it's a world leader in biotechnology, and I'll try to, in this presentation, to give you a range of examples to illustrate to you why I think that is a true statement. So this presentation is about the why we do at DSM what we're doing, what is driving us, what we're doing, how we're doing it, and then the now is giving you a list of examples of food application of industrial biotechnology. Well, I've got a presentation ready, but I'm also more than happy to divert in any direction. So if there's anything you feel I'm skipping or anything you would like me to elaborate, please interrupt me. I'm flexible, so let's tune the presentation to whatever you're most interested in. I'll show you later, DSM is not just about biotechnology. It is, I'll show you a timeline of our history, and we actually started as a mining company, so as black or gray as you can get it, and we still have a sizable part of chemistry. So it's not all about biotechnology. My presentation will cover the biotechnology in our company, and for a good reason. It's because I'll show you in that same timeline that as a company we made a tremendous shift over the past decades from a mining company to what is nowadays a nutrition company, where biotechnology is the most important discipline we have for producing our products. So why we do what we do? I think I've seen a couple of external presentations of the Bernal Institute, and I think some of the themes come back in your presentation as well. Yeah, we as science, we will have to answer a number of megatrends that we see developing these days, and a few of those trends that are particularly relevant for our company is the population growth. So 10 billion by 2050 is one of the projections. That's a sort of maybe the top of the population, after which it may decline a little bit. There's a lot of hunger in the world, but that number of hungry people is even exceeded by a number of people in the West that are overweight. So that you could say they also suffer from malnutrition, but then not quantitative, but rather qualitative malnutrition. There's a big issue with, well, we just had the issue with communicable diseases like COVID, but below that, there's still a bigger issue of non-communicable diseases like heart diseases, stroke, and cancer that are to a large extent also related to the quality of our nutrition. And then there's an environmental issue with an economic issue as well, with the amount of food that we lose and waste in the process of supplying our food. Yeah, so as a science-led company, we try to harness biotechnology as one of the main tools, as said, to tackle those challenges. And they are small, but you might recognize these symbols. They are part of a larger collection of symbols that are the Sustainable Development Goals that have been issued by the United Nations. And they're too small to read, but the 4 ones that we think we can particularly contribute to are zero hunger, good health and well-being, responsible consumption and production, and climate action. Those are the 4 that we picked out where we think that as a company we can make a serious impact on the world. So you see some of the topics there that are related to that. So it's feeding that growing population that I just mentioned in a more sustainable way with less impact on the climate, but also on biodiversity, on land use. We fight the climate change in terms of greenhouse gas emissions, and we try to prevent the overfishing of the seas, as I will give you one example about. So what do we do at DSM? We do a lot of biotechnology, and yeah, of course, biotechnology is a very traditional field. It's an artisan field where, if you look at historic evidence, for thousands of years, even more than 10,000 years back, people found evidence of beer brewing as one of the early forms of biotechnology. So it's been around. People have been using microbes to supply us with foodstuffs, to improve our foodstuff, to conserve our foodstuffs for years, using yeast, using fungi, bacteria, and algae as well. So they have been around, and yeah, we always tend to say that the microbes are the most agile and the most versatile chemists that we have around. So it's a matter of trying to harness their potential for production in our processes. So that's what we try to do, because from this more artisan approach of microbiology, what our company is about is industrial biotechnology. So that is using these microbes in the microbial fermentation vessels. Then we're talking about scales from 10, but usually 100 or more cubic meter scale, and do that in a well-controlled environment to have a consistent quality and safety of whatever we produce. So I promise you that timeline to illustrate that the tremendous the transition that we made, because you see the name of DSM, that is now the name of our company, that stuck somehow. That appears, but it's actually not the earliest part of this tree. The oldest part is the part in Delft where I'm working. That was not original DSM. That was started as the Dutch yeast and spirit factory, founded just over 150 years ago. So it's one of the oldest biotech companies in the world. It was producing at the time. It was founded by the first graduate of the Polytechnic School in Delft, which is now the Technical University, first chemical engineering graduate. And he knew that bakers were always sourcing waste yeast from the spirits factories, and that was what they were using for bread. So the yeast was a waste product, which didn't work very well. So he turned it upside down, and he thought, let's start making good yeast for bread, and that will be a higher quality product for the bakers, and then we'll get some spirit as byproduct. So he took the opposite approach. That's what started his company. And that is the company that is still there today in Delft, that part of the company. But as I said, there's a lot of acquisitions since then. So if you look in time, another important part of the current company is Roche. Well, Roche has still a very large pharmaceutical part that is a completely separate company. That's not us. We acquired a branch of Roche. That was Roche mostly about vitamins. So they produce a lot of vitamins, chemically initially, and later also biotechnologically. That got acquired. It got founded well over a century ago, but it got acquired in the start of this century, that part of the company. Well, DSM, as said, there was a mining company. Holland had some mining, but relatively small scale. So those mines They were closed in the late 60s because they were not profitable anymore. And at that time, the Dutch government, to prevent unemployment, they invested heavily in that company to retrofit it into a petrochemical company. And that company, because it was the largest part that acquired the other fishes, so fish eat fish, that was the biggest one. That's just the reason why the name stuck. But the name is officially the Dutch State Mines, and if there's anything we don't do these days, it's mining. So it's a meaningless abbreviation. But it stuck. It is now dominated, as said, by biotechnological activities that came from this early company, from the Roche vitamins business. And then you see a lot of the activities happened actually this century. We did a lot of acquisitions of an American company that I'll tell a little bit more about, Microbiome, working, for example, on a carotenoids platform. We acquired a company in the US, Martech, working on nutritional lipids, omega-unsaturated lipids. And we started a number of joint ventures, Avancia with Cargill, Olatin with a French rapeseed company, Ferromaris together with the German Evonik company. And then very recently we did some more acquisitions, and we also started another big partnership with Delft University on artificial intelligence. So a quite dynamic history. And as said, we ended our mining activities in the late 60s, turned into a petrochemical company to retain employment. There we turned from petrochemistry into fine chemistry, and now from fine chemistry we morphed into a company that is getting by far the largest profits out of nutrition. So complete transformation over the years. So the current strategy, and this is a presentation that was really released in February of this year, so it's fresh. So this is the way our board currently looks at what we're here for. People always like these repeated letters, so they found 4 P's that describe what we do, and the 4 P's that they have found relate to pathways. So pathways is about, because we know a lot about chemistry and biochemistry, we have the toolbox to combine, to also make hybrid processes where we combine microbial conversions, chemical conversions, recovery processes, to make a lot of different molecules, nutritional molecules, different classes, for serving as food and feed ingredients. The proteins, Luc already alluded to that. So our applications are food and feed production, and of course, protein is one of the bulk ingredients of a lot of food and feed. It's also one of the ingredients that puts a lot of pressure on the environment, with, for example, fish meal, pressure on the biodiversity in the seas, replaced by soy meal, but soy meal puts a lot of pressure on the Amazon. So can we find new pathways to produce more sustainable proteins? Prevention is more on the nutraceutical side of things. So can we make ingredients that really help prevent diseases? I'll come back with examples later, but think about ingredients that improve the gut microbiome to prevent rather than cure diseases. And precision, that's the latest activity. It's about novel ingredients, but it's also a lot about digital solutions, trying to personalize nutrition, find out what people have too much in their diets and what they lack of, and how you can tune diets to what people need. And that links back to what I said about the wasting of food. Yeah, if you eat what you don't need, then you waste it in the end. So it's not just about what you put in your garbage bin because you left it too long in your fridge. It's also whatever you eat, what you really don't need. So can you tune your diet to your personal needs? Yeah, so what we do is industrial biotechnology in the broad sense, and we try to stay well tuned to the external developments in that field. So there's a couple of key developments that we have integrated in our R&D facilities in-house. Of course, since the 80s already, the traditional ways of recombinant DNA technology, but I think you're all well aware of for example, the CRISPR-Cas revolution of the last years, of really gene editing, tailor-made modifications, which is nowadays it's really a standard tool in any microbe that we can use, for which we can use GMO technology. Of course, there's always foodstuff that still remains non-GMO, so that we have to resort to the more traditional ways of strain improvement. But for quite a lot of molecules, we can apply the modern GMO technologies. Mathematical model. Well, I hope that the 4 students that I've been talking to for the past 2 days recognize my message here, that we think we can accelerate our R&D work by doing a lot of modeling, supporting our experimental work. This is the famous DBTL cycle, the design, build, test, and learn, where you don't just go to the trial and error phase of setting up an experiment, seeing it failed, and trying to do better than the next time, but you do it explicitly. You do a learning cycle where you first tell what you expect from an experiment, because you always have a first model in mind. You just have to make it explicit. You build your installation, you do the experimentation according to the design, and you explicitly learn, because your model will always be wrong, you always mispredict it, but you can use that mismatch to improve your model and run that cycle. So that is really explicitly done nowadays, not just for process improvement, as I've been teaching here for the past 2 days, but also very importantly for strain construction. So we construct strains with these modern tools, with the CRISPR-Cas, but also we analyze strains. We do a lot of omics analysis of the strains that we make, metabolomics, proteomics, transcriptomics, where once we have fermented our strains, we measure what the physiology of the strains looks like on those various omics levels, and we see whether the changes that we made actually turn up in the phenotype of our strains. And if they don't, we close that cycle and we try to learn how we can make better constructs in the next round. And in order to do that, then, if you can synthesize, you have the CRISPR-Cas and you have the model to design it, you also need to increase the throughput of your labs. So we try to robotize what we can nowadays to increase that, the sheer numbers of strains that we can make. Yeah, so this is I guess a little bit more higher resolution, some of the techniques that we use. So I mentioned we still have the old-fashioned screening. We have to, sometimes for some products we have to screen for the best host strain. Sometimes we start making completely new classes of molecules, and then you have to spend your time well in looking and thinking about the best host to use. It's not always the standard bug you have on the shelf that is the best host for a new molecule. So you really have to think, is it a fungus, is it a yeast, is it bacteria, and which of the ones would be most suitable for this product to make. That's a very important design at the start. So we got a big own strain collection that we can source from. Then the strain development, so especially for the products where we can use the GMO technologies, we've got these techniques that I just mentioned, where we can tune the pathways to our needs. Well, the field where I'm mostly responsible for in the company is bioprocess development. So once you have the organism, of course you have to make sure that you create the conditions where it does whatever you want it to do in a large scale, in those big vessels where conditions are not always homogeneous. And so you have to master that, because for R&D, our work is done when the factory is running. So tech transfer from the lab to our plants across the world is still part of our responsibility. Yeah, and then some supporting technology, the more traditional ones I would say is biochemistry and microbiology. So really make sure you have the molecular level insight in the microbes. I mentioned the omics technologies. You need the data side of things. So you need to be able to that generates a lot of data. You need to be able to handle that and turn the data into information. Analysis, this whatever is on top of those technologies. This is more the classical analysis, just characterization and quantification of molecules. And then data science, this is really the basics of statistics, of big data handling. And I mentioned in the example of the recent collaboration with Delft University, also artificial intelligence. So try to have the computer support you in turning data into knowledge. So there was a diversity in techniques that we use. We also have some diversity in locations where we do this. This is limited to just our biotechnological R&D locations. So this is not production sites. The places where we do R&D there are 8 of them, and that is because of the timeline I showed you that DSM has grown, not organically, but mostly by divestments and acquisitions. So all of these labs actually came in by acquisitions, and all of these labs, one of my challenges is to try to synchronize some of the methodology that we use, but at the same time, each of them came with a specific competence on the product that they were making. So they also are all unique in some of the product-specific competences. So we have 2 labs on the East Coast in the US. This is the lab that I'm working in Delft, but we got a smaller lab also at the Agriculture University site in the Netherlands, a site in Denmark, Germany, Austria, Switzerland, and one in the Eastern China. And the other dimension of the collaboration is not just internal collaboration between the labs, but I have shown you a couple of joint ventures that we started over the past decade. So I mentioned specifically the ones. This was that rapeseed company in France, in Cargill. That is our Avancia sweetener joint venture. And with Evonik, we're producing this omega unsaturated fatty acid as salm feed. But you see there are more academic ones as well as companies that we collaborate with. Yeah, as a finish of what we do, this was all about technology. We also realized that there's a societal aspect to biotechnology. So it's not just about what you can do, but it's also about whether the general public wants you to do what you do, whether it's considered as the path to go to. So what we do is we try to advocate also the responsible use of biotechnology. So we try to explain why we think that for a number of products, biotechnology is the way to go, including GMO technology where needed, because not everything can be done with the classical technologies. So we partner up with various international organizations to try to bring that message across of why we truly believe that for a sustainable future, you will need biotechnology as one of the tools in the human toolbox. Yeah, then after the why we do it, what we do and how we do it, this is what we a selection of products that we're doing now. So this is trying to give you a flavor. I introduced the history of biotechnology with bread making and yogurt making and beer making. That's a little bit of a cliche by now. So I try to. I hope that all of you will find some products that you didn't think of before. And this is why I think that why we are a quite exceptional company in terms of biotechnology. It's because of the width of the portfolio that we have. It's not exhaustive, so it's not listing all the products, but it's listing a diversity of the products. And if there's anyone that you want to dwell some more time on, just interrupt me. So one of them is what we call Maxillact, or market as Maxillact. That's a lactase. It's an enzyme. We make a lot of food enzymes, so enzymes that we supply to the food industry as a processing aid. There will be more examples. Maxillact is one of the examples. So a lactase is used in dairy products. It converts lactose into sugars. So there's lactose intolerant people that need products without lactose. At the same time, what you do with lactose is you can attain sugar reduction in the diets. So that's fighting that obesity that I mentioned, because the lactase will cleave. Lactose is not sweet. It's not perceived as sweet. If you cleave it into the separate molecules, then that creates sweetness in the food. So actually, you don't add sugar to your food. You just you split the sugar that makes taste perception sweet. So it's a great way of not adding sugar and still increase the sweetness of products. Yeah, putting the life in yogurt. So this is about not producing enzymes, but producing the microorganisms as our final product. So the living microorganisms. Delphipro is one of the products. We make the traditional lactic acid cultures, so acidifying to produce the yogurts and similar products. Also some related probiotic strains that both for animal and for human feed. And as I mentioned, that this is linking to that P of prevention. This is typical food stuff that is on the rise to modulate your gut microbiome, to bring it in a more healthy balance. And that's one of the demonstrated effects of good microbiomes is, for example, is immune system boosting. That's one of the ways to increase health by this sort of ingredients. So Maxorom. Maxorom is a trade name of another product. It's not a protein. It's not a live culture. It's an extract. It's a yeast extract. So that's what we do also in Delft. There we have a factory. We produce all yeast. It's not a traditional baker's yeast. It's a special family member that contains a lot of taste enhancing components. So this component has been around for a long time. Yeast extracts are well-known ingredients for soups, for sauces. So it's a savory kind of taste that you add to it. A big new market for this product is suggested by this one, the meaty taste. So, for example, in meat replacement, the new vegetarian meat products, this is one of the components that helps create a more meat type of flavor, and to mask some of the off flavors that some of the meat replacers tend to have. Yeah, yet another class of molecules. So I've talked about the enzyme, I talked about live cultures, I talked about extracts of whole cells. This is a small molecule. This is that joint venture that I mentioned a couple of times now with Cargill. So Cargill does the downstream processing and does the marketing of the product. We do the upstream production of it. It is stevia. Traditional stevia is an extract, plant extract, but again, you need quite a lot of surface area to grow these plants because the concentrations of Reb-M are low. We use microbial cultures that produce the same molecules in a much more intense manner. So we produce the riboside molecules, that is the chemical name, that are in stevia as a low-calorie sweetener. So again, fighting obesity as a nutritional issue, fighting land use, because we need less land use to produce the same amount of this sweetener by having the microbes overproducing this stuff rather than the low concentrated plant leaves. Yeah, back to enzymes as products. Purifin is an lipase. Oil industry has one step in oil processing which is called degumming. A lot of fatty fats contain some phosphor groups, so you get a couple of fatty acid chains on the glycerol, but sometimes some phosphates that you need to cleave off because they lead to gum in oil products. We have lipases that cleave off that phosphate group, and so they are used in oil processing, and they give yield benefits for the producer. So again, it prevents waste in the production of the food chain. Yeah, when I was explaining that the timeline of DSM, one of the acquisitions I mentioned was this Microbiome company in the US. So Microbiome got acquired because of its knowledge on the carotenoids. Carotenoids is a class of molecules that has two main functions. It's an antioxidant, and it's a coloring agent. So there's multiple forms of carotenoids. Not easy to produce. They're not as big as enzymes, but they're still complex molecules. So this was typically a quite expensive product, and with a lot of chemical synthesis steps needed. So what Microbiome did and what we further developed after the acquisition is a platform for producing a number of these carotenoid products microbially. So just one step, the microbe excretes the right carotenoid, and that is what we now start to market for food and beverage applications. Anyone so far who knew all these biotechnological products? See if I still meet my target of teaching you well. So this is HMO, human milk oligosaccharides. Yet another class. So it's a sugar type of class. If you look at mother's milk, if you look at the weight fraction of the various components, then HMOs are the third class in terms of size, from large to small. And these are it's in a class of molecules because there's a lot of variants with different sugar molecules connected. But they are generally known to have also an effect on the microbiota of newborn. So that's why it's a mother's milk, and that's why anyone who's selling baby milk powder, so artificial mother milk, they want to mimic the natural milk as good as possible. So this is what a lot of those companies. We are not making mother's milk. We are not selling. We are not B2C. We are B2B. We are supplying the business. So in this case, the infant milk companies we're supplying with the ingredients. They are asking us for this ingredient to have that health benefit of their product. Vitamins. I told you about the acquisition of Roche vitamins in Switzerland. Vitamin B2 is a famous example of one of the vitamins that was traditionally also always synthesized chemically. So Roche was great. A lot of vitamins are also quite complex molecules, requiring a whole train of conversions, purifications, chemical conversions, that is, so catalytical reactions. I think most of you know that if you want to do this synthetically, making complex molecules, there's a lot of activity. You have to block groups, then specifically react non-blocked groups, get rid of that blockages. So that always took a lot of reaction steps, made the molecules costly, but also created a lot of chemical waste in using solvents, in using catalysts, metal catalysts. So vitamin B2 is one of the examples where they were able to successfully clone the genes into bacillus in this case, and have a one-pot fermentation process where simple ingredients, glucose and some salts, are converted into the final product in one step, so vitamin B2. So that's one of the examples where chemical processes were converted into biotechnological processes to really decrease the impact, in this case energy cost, byproduct effects on the environment to mitigate those aspects. Yeah, so microbiota, I mentioned probiotics before. That earlier example was for human feed, but we have a big part of the company focused on animal feed. So for animal feed, we also supply probiotic cultures as well as combinations of vitamins and trace minerals. And I mentioned in the current strategy, I mentioned that last P of precision. This is one that links to that precision, especially in that animal feed producing. What we do a lot of data analysis. So we have a lot of partnering with farmers, with animal breeders, where we analyze what the animals eat, how their health status is, how fast they gain weight, how healthy they are, what they excrete, and we collect all that data and use that information as big data streams to tune the diets to the species and even sometimes to the subspecies level. So not one feed for all the pigs, but really tailored to young pigs, to older pigs. So it's diversifying and really tailoring it to the needs of the animals. Again, it's for the health benefits of the animals, but it's also because if there's any impact of food production on the world, animal farming is one of the bad guys. There's a lot of waste, ammonia waste, greenhouse gas emissions related to that, and that can partially be mitigated by better tailored feed solutions. So Luke, I hope that you didn't know this one yet. It's one of the latest products that we have. It came in also by an acquisition of an Austrian company who are specializing in the mitigation of mycotoxins. Mycotoxins are typically protein peptides produced by fungi, and fungi grow in animal feed. They grow everywhere, and yeah, sometimes these mycotoxins, it's a class of molecules, sometimes it's present without you knowing it. Over the years, they have discovered more and more mycotoxins that actually go at the cost of animal health. And what this company that we acquired a few years ago has developed is a portfolio of enzymes that specifically break down these toxins. So they've been screening for those enzymes that cleave the toxins into innocent compounds, breakdown products, and thereby have better animal health. Did you know that one?
I know the problem, but I didn't know the solution.
Okay. I think this is the final one for today. So I hope then that I give you a neat impression of the width of the food and feed portfolio, or the ingredients for the food and feed that we are making. Saccharomyces, it's one that's close to my heart because I spent quite a few years, the last years, on this particular project. It's omega unsaturated fatty acids. Two of the major ones are DHA and EPA. And if you eat salmon, salmon has a health claim because it contains these omega unsaturated fatty acids. They're good for eye development, for brain development, and work against heart diseases. So there's some proven effects of these fatty acids. Well, the salmons don't produce these fatty acids. They just have them because they eat them. They eat fish. Those fish, they don't make them. They have them because they eat algae. So that's the whole food chain in the oceans. And a lot of the salmon nowadays is not wild catch, but it's bred in Norway, in Chile, in Scotland. And they are fed with wild catch in the Pacific. So the Pacific has an overfishing issue these days to provide just the fish meal and the fish oil to feed to the salmon in those cages in Norway. That is hitting the limits of the ecosystem, even an ecosystem as large as the Pacific. So what we're doing in this project is that we short-circuited the food chain. We don't feed the fish to the salmon. We feed the oil. Of course, it has to be formulated in something that the salmon will want to eat. So that the salmon feed producers, they use our oil, they formulate it, and our oil then contains these unsaturated fatty acids. So we developed an algal strain that produces it, and we developed a bioprocess to grow those algae and to purify that oil for the producers. So then trying to prevent that biodiversity issue of overfishing, as well as the health benefit of the omega unsaturated fatty acids. Because one of the things that was happening because of the decreasing stocks of fish oil was actually that the salmon industry was decreasing the amount of fish oil they put in their feeds to the bare minimum levels still to meet the specs of salmon. If you compare nowadays salmon with salmon of a few decades ago, you will find that these oils have decreased quite a bit in salmon just because the salmon breeders have gone to the low limit of their specs still to have the health claim. So that's of course what you don't want to continue. So that was the last example, and that was my story for today, but I'll be more than happy to answer any questions that you have. Yeah, I hope it gave a little bit of insight in what biotechnology can nowadays do in the realm of food and feed production. And yeah, thanks for your attention.
Thank you. Maybe to kick off questions and then leave them for the rest. One of the topics that I introduced you with was the alternative protein production, cultured meat. You have significant press releases in the last period. We host the Dairy Processing Technology Center, which is the processing arm of the conventional industry that is suffering from significant pressure to reduce their footprint. And at the same time, we have the cattle industry where for the meat production side of the house, where the same problem rests. Alternatives are getting to the market. Some level of discussion is on cultured meat. For this institute, we are looking for the future of biopharma in particular, and their protein production is very central. But developments are certainly also in the stem cell development, stem cell for personalized medicine applications and so on. It's essentially exactly the same technology as you would be using for cultured meat. So there's a lot of technology that would link to, for instance, the pharma developments as we have them here. Could you elaborate a little bit on how you see that development, and also where the strengths of this institute, which are on the product side, on the material side, making that into a solid product, the low footprint, that's would be connected to this sort of sectors and so on. Where do you see the interfaces?
Yeah, the protein work that we're doing, to give a little introduction on that, I've mentioned a couple of proteins, but those are enzymes, so that are some of our current protein products. One of the directions we're currently taking into more bulky proteins, so really as the bulk ingredient for feed and food, is I mentioned the joint venture with Avril, so that is the rapeseed company. It's a nice example of reducing waste. What Avril has always been doing is producing rapeseed oil, and the residue of that is pressed cake. So what we added onto it is that we extract protein from that pressed cake because it's a functional protein that's in there. So trying to turn waste into a bulk protein that can be used for food production. Well, that is great. If you have some waste of protein, well, try to that is the spirit of biorefining that you hear a lot about these days. Don't call it waste. There's always something to gain from it in a cascading approach. So that's the first thing that we look at. If there's any opportunity to gain proteins from such strains, that has a low environmental impact. Longer term, what we're looking at is, well, DSM is one of the many companies, I think, across the world that has committed to net zero emissions in 2050. I think that a lot of boards of directors have committed to that without really thinking about what it actually means to be truly net zero. And yeah, it's the honorable task of me and my colleagues to then translate that commitment into reality. And to be honest with you, when we started seriously thinking about the consequences of net zero food and feed production, we were a bit shocked about what we'll need to do, because it means literally zero fossil inputs. If you think about the food chain, how many places fossil inputs are currently used, then a bio-based future is not going to bring us to net zero. Because, as an example, if you think currently at agricultural practices, of course, sugar is bio-based. The crop fixes CO2 to produce that sugar. But before that sugar enters our gates as a fermentation feedstock, already about between half and 2/3 of the CO2 that was fixed in the sugar has been released in a fossil shape because of agricultural practices, sugar refining practices. Well, and then part of our sugar is turned into CO2 by our organisms. We have the downstream processing, we have the transporting, and then the food is consumed and it turns into more CO2. So by the end of it, that bio-based foodstuff has released 2 or 3 times the amount of CO2 that was initially fixed by the plants. So net zero, no way. So thinking about that consequence of making it truly net zero, we see no other way of turning carbon dioxide and renewable energy into foodstuff, because that CO2 all needs to be fixated. That ends up in the foodstuff that we breathe again if we have digested it. And so it's a very long introduction to my answer to you, that we think in the end that biotechnology is not about just turning sugar, the traditional biotechnology sugar, to whatever I have just been presenting in terms of molecules. Longer term, we will have to turn renewable electricity as the energy source, as CO2 as the carbon source, into all of these molecules. And then it may be partially agriculture, but there will also be a lot of other types of conversions of CO2 and energy into our feedstocks. And that includes processes, that includes sustainable energy production, a huge amount, multifold of what we're using these days. So if you talk about your windmill industries, anything going in that direction of sustainable energy production, highly relevant for that. But also conversion technologies of all industries making whatever feedstocks you can use. Think about ethanol, methanol. You need the right materials for those conversions. Those will not all be fermentation processes. They will all be, we'll need all tools, include some electrochemistry and the right materials to run those processes. So I think that upstream part, so it's upstream for fermentation, I see that is still, for a part, it's still low TRL. And I think that there are institutes that have materials knowledge as well as biotechnology knowledge are very well positioned to answer some of those challenges ahead.
One of the reasons that we have the minister here later today, because of the wind ambition that is probably from the estuary, significant carbon emissions from, for instance, Irish cement. So the biggest point sources for renewable carbon are here, and the need for connecting also to food, but also transport industries. So the sort of thinking behind it. Questions?
Thank you for a very interesting talk. I didn't know half of those things that DSM made.
Oh, great.
So if you were, I'm just wondering what DSM's approach is. Like, for example, in pharma, the pathway or the developmental pipeline is, you know, we know where the bottlenecks are and where the expense comes in research and development. In your processes, like, I'm just wondering what DSM's approach is. Like, if you have, say, a target protein that or enzyme that you want to produce for one of your applications, where is the biggest challenge? You know, is it in screening the high performance organism? Is it in the purification later on? Is it in, like, in general, where do the failures happen?
Yeah, great question. I think very traditionally it's a matter of beating competition. We're one of the enzyme producing companies. What I like about the DSM is the width of its portfolio. You can see a downside to it is that if you do a lot, then you're not focused on anything in specific. So that is always a challenge for us. I can mention the famous example of Novozymes. Novozymes is very, very good at producing enzymes, and they're thinking about it all the time. Whereas we, as I showed you, we're thinking about all kinds of products. So that is a big challenge, being ahead of Novozymes, because they're almost as big as we are, a bit smaller, but focused on enzymes only. So beating them to it is not always easy. Sometimes it's helped because we're so broad. We just see niches that they are not aware of. So they are great at what they do, but I think we're maybe a little bit more creative at finding niches. So that's a challenge. It's a sheer business challenge, finding the new enzymes where the world, the market has not been conquered by others yet. And second one, I think, what I see, but maybe I'm a little bit biased on the processing side, is that the genetic tools have really taken off over the last decades. So finding a protein with a functionality in the databases, finding a class of lipases, well, you can find a lot of them, but with the robotics and with the precision tools, getting those codes, having them just the genetic constructs produced for you to clone into the organism, that's no longer the bottleneck. That is really pushing the button almost. So constructing the strands is not that difficult anymore, but still, particularly for enzyme that you ask about, proteins are sometimes hard to express because it's not just about making that chain of amino acids. It's about the folding. Sometimes it's about functionalizing that chain, excreting it, and that is not fully controlled yet. So we just do have strains that have the perfect genetic construct. Sometimes they even express it intracellularly, but don't excrete it. Some of those enzymes need metals to be active, and we don't get the metals in the active spots. So it's in the end coming from a good construct, genetic construct, to a strain that actually overproduces it in commercial quantities on a larger scale. That's that step of getting that construct, giving that high titer in fermentation in practice. That is in my experience, that's the hardest part of protein production development. Yeah.
Thank you.
Thank you. Yeah. Other questions? not at this moment. There's a lot of digesting taking place, and that is what we will be doing also in a moment in our lunch, for which we have to go. Maybe just as a slight answer to that, standard in biologicals manufacturing at this moment, monoclonal antibodies. Many of, I think about 10 of the 10 pharma companies produce monoclonal antibodies. We are still at relatively high cost levels, meaning that the cancer therapies are still, also due to the cost of the monoclonal, around 70,000, 150,000. Breast cancer therapies around around 100,000, making it impossible for the rest of the planet to do. So being able to significantly reduce cost using this sort of precision technologies, which a conventional company would not directly go into, but would enlarge the market significantly. So we once were engaged in a Unilever project where monoclonal antibody targeting, the stuff that is in your mouth, added to toothpaste, could be much more efficient in killing the bugs that you don't like. But there's no way you can pay that amount of money for it. It's the same monoclonal antibody. So cranking up the titer, which from a pharmaceutical point of view maybe not be that pressing, but for this sort of application would open a completely different market opportunity also.
Yeah, these techniques I showed, they have been for a good reason. Some of them, like the CRISPR-Cas, have been awarded in recent years with the Nobel Prize. They are really a revolution. My first big project in DSM was a strain improvement project for penicillin production, the old product. And we had genetic tools for the first time we were applying them. And it took me 3 years to make 20 mutants. So 20 individual enzymes overexpressed or knocked out. And I selected them on a big literature study to have overproduction of penicillin. And that took me 3 years. And in the end, none of them made it to the factory. And that was between 2008 and 2010 that I ran that project. And now we are just over a decade later. And they just laugh at what I did at that time. They literally do now in a month what I did in 3 years. So that is not to be underestimated. What power that unlocks. If anything can be called a revolution, it is this. And then compared to that was the background of my answer. Process development is still relatively slow. It has not seen the revolution that strain construction has seen. So yeah, a little bit of what I've been teaching these days of